Assalamualaikum. I'm Asiya Haleema Unar, lecturer of Botany. Today I'll deliver a lecture over germination. First, let's talk about what the germination is. It is the condition of breaking of dormancy of the seed to produce the seedling. Or in other words, we can say that germination is a process by which the embryo in the seed grows into a new seedling. What are the types of germination? There are three types: apigeal, hypogeal and viviparous. I'll elaborate each of them in separate section. First is apigeal germination. The word apigeal is divisible into two. Epi means above, geos means earth. Now in this type of germination, the hypocotyle part elongates rapidly and arches upward, pulling the cotyledons which move above the soil. Then they become flat green or leaf-like in appearance. For example, the bean, the cotton, the castor oil seed and the papaya. Now this is the diagram which shows the apigeal germination, that how the germination occurs above the soil or above the earth. Second, what is hypogeal germination? Hypo means below and geos again means earth. Now hypo is the opposite of epi. It is a kind of germination during which the cotyledons are seen to remain inside the soil due to the rapid growth of the epicotyle part. For example, the maize, grain, pea, gram and etc. This diagram shows that this is hypogeal germination occurring below the earth. The last one is viviparous germination. It is a type of seed germination in which the seed germinates inside the fruits while still attached to the parent plant and simultaneously is nourished by it. The radicle elongates, swells in the lower part and gets stouter due to the increasing weight of the seedling. In the last, then it separates from the mother plant. For example, the palm, the coconut, the rhizophora and etc. This diagram shows the viviparous germination. Now, what are the requirements or the parameters needed for germination? Namely, there are three: air, water and optimum temperature or light. Now, I'll explain those three parameters, that why they are necessary through an experiment which is known as 3-bean experiment or 3-bean seed experiment. Now this is a 3-bean seed experiment. It shows that 3 bean seeds are tied on a glass slide and keep this slide in water containing beaker in such a way that one seed is completely immersed in the water, the middle one is half immersed in the water and upper one is not in contact of the water, it's just in contact with the air. You see in the experiment, the last one is the completely immersed one, the middle the half immersed and the third one is not immersed. After a few days we observed that germination has taken place in the middle seed. Why? Because this seed gets all three parameters needed or required for germination, that is the air, water and the temperature. Why water is necessary and why light is necessary? Now take some gram or pea seeds. They are put on a dry cotton wool in a beaker A, and same quality seeds are put on moist cotton wool in beaker B. You see in the diagram, beaker A and beaker B both are shown. After 2 or 3 days, we observe that there is no germination in beaker A while seeds germinate in beaker B. This demonstrates that why water is necessary. The question may arise, why? The answer is that food is stored in the seeds in dry condition, but the developing embryo cannot utilize this dry food, so the food can only be utilized in liquid form and only seeds can utilize this one in the dissolved oxygen. Further, excess water stops the germination because once all the dissolved oxygen is utilized by the seed, further germination is not possible as the life supporting oxygen is now lacking. Why air is necessary for seed germination? Again, I'll explain you in terms of a diagram. Some seeds of gram or pea are placed on wet cotton wool in beaker A, that's shown in the diagram, beaker A. Some water is boiled in beaker B to expel out the dissolved air which is already present from the water. The hot water is then cooled and similar gram seeds are put in it. A few drops of oil are also put in beaker B to prevent water surface that will prevent entry of atmospheric air within two. It is observed that germination has taken place in beaker A but not in beaker B. Now this experiment proves that air or oxygen is necessary for seed germination. During germination, rapid cell division takes place and energy is required for cell division. Okay, this energy is obtained from the oxidation. The oxygen required for oxidation is supplied by the air. So thus air is also one of the important parameters for germination. Now the next topic is transition to flowering stage, that is from seed to seedling. So there are three main steps of seed germination into a seedling. The first is the seed germination. In the seed take up water rapidly and this results in swelling and softening of the seed core at an optimum temperature. Of seed coat help to immerse the radicle and the plumule to form a primary root. Now this stage is referred to as an imbibition, simply taking up the water and swelling from itself. In the second step, the internal physiological activities starts to respire and produce protein and metabolize the stored form of the food. This is the large phase of the seed germination and this is the final stage of seed germination. In this stage, the cell of the seeds are elongated and divided, which brings out of the seed, and cotyledons are expanded, which are the true leaves of the new plant. Now I explain you in simply terms of the diagram. This is number one, the seed soaks up water, means the seed starts to get water into itself and swells. This is imbibition. Seed coat splits. This is the second diagram. After getting water, now the embryo root grows out to get the water. Again the embryo root comes out of the seed and it gets further water. Then embryo stem grows. Finally the leaves grow on the stem. Then the seedling can make photosynthesis process by itself. Then flowers are formed. Okay. Now thank you very much.